Bismillahirrahmanirrahim. Welcome back to my channel. In this video, I'll discuss the basis of generation of electrical activity in the excitable cells. The plasma membrane is selectively permeable, which allows a cell to maintain different concentrations of certain substances on either side of the plasma membrane. A concentration gradient is a difference in the concentration of a chemical from the inside to the outside of the plasma membrane. Many ions and molecules are more concentrated in either the cytosol or the extracellular fluid. For instance, sodium ions are more concentrated in the extracellular fluid than in the cytosol. The opposite is true for the potassium ions. The selective permeability of the plasma membrane also creates a difference in the distribution of positively and negatively charged ions between the two sides of the plasma membrane. Typically, the inner surface of the plasma membrane is more negatively charged and the outer surface is more positively charged. A difference in electrical charges between two regions constitutes an electrical gradient. The combined influence of the concentration gradient and the electrical gradient on movement of a particular ion is referred to as its electrochemical gradient. The electrochemical gradients are important because they help move substances across the plasma membrane. In many cases, a substance will move across a plasma membrane down its concentration gradient. Similarly, a positively charged substance will tend to move toward a negatively charged area, and a negatively charged substance will tend to move towards a positively charged area. Voltage and current are two important factors to consider in the study of electricity. Voltage is the measure of potential energy generated by separation of charge. It is measured in volts or millivolts. The greater the difference in charge between 2 points, the higher the voltage. Voltage may represent a source of energy, also known as the electromotive force. The flow of electrical charge from one point to another is called current. The amount of charge that moves between 2 points depends on two factors, namely voltage and resistance. Resistance is the hindrance to the flow of charge. Since there is a slight difference in the number of positive and negative ions on the two sides of the cellular plasma membrane, there is a potential difference across the membranes. This potential difference leads to a voltage called the membrane potential. In electrically active tissue, the potential difference between any 2 points can be measured by inserting an electrode at each point and connecting both electrodes to a specialized voltmeter. Diffusion occurs due to the tendency of particles to redistribute from regions where they are highly concentrated to regions where the concentration is low. Additionally, the difference in potential established between the outside of a cell and the inside of the cell also affects the flow of current across the plasma membrane. In the body, electrical currents reflect the flow of ions across cell membranes. Alterations in membrane potential are very important since these are used as electrical signals in the nervous system. One of the most important alteration in membrane potential is the action potential, which serves as an electrical signal in the body. Membrane potential and action potential generation depend upon movement of ions across the cell membrane governed by electrochemical gradients. Ions are charged particles and are hydrophilic. Hence, these cannot pass through the cell membrane without assistance. Now the question arises that how will the ions move across the membrane to produce a separation of charges, also known as the membrane potential? The answer to this problem is provided by the transmembrane proteins. These include passive transport channels as well as active transport pumps, which are necessary to generate a transmembrane potential. If you are interested in mechanisms of passive and active transport across the cell membrane, do watch our videos on the topics having links in the description. Ion channels are pores that allow specific charged particles to cross the membrane in response to an existing concentration gradient. Some ion channels are selective for charge. These channels allow cations, particularly sodium, potassium, and calcium, to cross the membrane but exclude anions. This is called electrochemical exclusion, meaning that the chemical pore is charge specific. Channels for cations will have negatively charged side chains in the pore, and channels for anions will have positively charged side chains in the pore. Ion channels can also be specified by the diameter of the pore. This is called size exclusion. Moreover, a channel may have several different states corresponding to different conformations of the protein. But each state is either open or closed. In general, closed state of the channel restricts movement of the ions. When a channel is open, ions permeate through the channel pore down the transmembrane concentration gradient for that particular ion. The two main varieties of the ion channels are leak channels and gated channels. A leak channel randomly alternates between open and closed state. There is no actual event that opens the channel. Instead, it has an intrinsic rate of switching between the open and closed states. Typically, plasma membranes have more potassium ion leak channels than sodium ion leak channels. And the potassium ion leak channels are leakier than the sodium ion leak channels. Thus, the membrane's permeability to potassium is much higher than its permeability to sodium. Leak channels are found in nearly all cells, including the dendrites, cell bodies, and axons of all type of neurons. Leak channels have important contribution to the resting membrane voltage of the excitable membranes. Ion channels do not always freely allow ions to diffuse across the membrane. Some are opened by certain events, meaning the channels are gated. Gated ion channels are found primarily in the cells of nervous or muscular tissue. Gated channels can be further categorized on the basis of how they are gated, or in other words, they are categorized on the basis of the event that switches them between open and closed states. A ligand gated channel opens and closes in response to the binding of a ligand or chemical stimulus. A wide variety of chemical ligands include neurotransmitters, hormones, and particular ions can open or close ligand gated channels. The neurotransmitter acetylcholine, for example, open cation channels that allow sodium and calcium to diffuse inward and potassium to diffuse outward. Ligand gated channels are located in the dendrites of some sensory neurons, such as pain receptors, and in dendrites and cell bodies of interneurons and motor neurons. A voltage gated channel opens in response to a change in membrane potential or voltage. Voltage gated channels participate in the generation and conduction of nerve impulses in the axons of all type of neurons. A mechanically gated channel opens or closes in response to mechanical stimulation in the form of vibration, such as sound waves, touch, pressure, or tissue stretching. The force distorts the channel from its resting position, opening the gate. Examples of mechanically gated channels are those found in auditory receptors in the ears, in receptors that monitor stretching of internal organs, and in touch receptors and pressure receptors in the skin. As described earlier, membrane transport of ions is necessary to generate a transmembrane potential and an action potential. Of special interest is the carrier protein referred to as the sodium-potassium pump that moves sodium out of a cell and potassium into a cell against their concentration gradients, thus regulating ion concentration on both sides of the cell membrane. This pump moves three sodium ions to outside and moves 2 potassium to the inside in each cycle. This creates an imbalance of charges where there is a slight excess of positive charges on the outside of the membrane and excess of negative charges on the inside of the membrane. This slight imbalance of charges sets the stage for generation of action potential in neuron and muscle cells. Hence, sodium-potassium pump is also known as an electrogenic pump. Thanks for watching and listening. Please do share this video with your friends and colleagues. If you like this video, please subscribe to this channel and watch more videos.